Thank you, Dr. Watanabe. Your overview of the Ajinomoto's robust conjugation platform shows how chemistry innovation continues to drive reliability and versatility in ADC productions. And it's a great reminder of how strong process engineering supports the entire ADC ecosystem. And thank you again to all our speakers to your insightful and thought-provoking talks. We now move into our panel discussion. So please, for our audience, please continue to submit your questions and comments through the Q&A box. We look forward to addressing them. And now I'd like to invite my co-host, Dr. Qing Li, to kick off the discussion. And Dr. Li, we now turn it over to you.
Okay, yeah, thanks, Inxi. And hello, everyone. Now, welcome to our panel discussion session. And we are really fortunate to have with us like four experts on, all this innovative site-specific conjugation platform. So now let's dive into the science. I'd like to start with the topic that's critical for any ADC program, which is the payload. So let's discuss about the payload compatibility. Are there limitations on the types of linkers or payloads, thinking about factors like the size, charge, hydrophobicity, that can be used in your site-specific conjugation platform? And how do you screen for those optimal payload linker combination? Dr. Merrily, perhaps you could start from your perspective.
Yeah, so I don't think there's a limitation, like a physical limitation for combining linkers and payloads. I think what's really important is to pick your payload based on the disease that you're trying to address. Not all cancers are susceptible to some toxins, and it's important to pre-screen your tumor types for the correct payload. So, for example, heme tumors tend to not be as responsive to topoisomerase inhibitors as they are to microtubule inhibitors. So perhaps for more efficacy, we need to move towards a certain class of payload.
Okay. Yeah, thanks, Macho. So, does anyone else want to add their experience with payload limitation and screening strategies? Okay. Okay, great. So that's brilliant. And now a brilliant conjugate in the lab is only useful if it can be manufactured at scale for clinical use. And so the next question will be about CMC. Could you please share the journey from milligram scale research conjugate to a kilogram GMP batch? And for your specific technology, what is the single most challenging step to scale up? Dr. Watanabe, so from this AgiCap standpoint, what are the key scale up hurdles you had to overcome?
Yeah, thank you. Yeah, actually, I have experience from the milligram scale to gram scale preparation under GMP. And from our technology, we have very good reproducibility from the milligram scale to gram scale. The only difference is the purification method. And we use gel filtration and centrifugal filtration in small scale. And sometimes it causes the decreasing the yield to sticking the membrane. But this is not our technology specific issue. So, and the scale up, we use TFF operation and we improve a little bit the yield, around 90%. So from our, I believe our technology is very reproducibility from the scale up.
Okay, yeah, thanks, Dr. Watanabe. So, Dr. Fan, like, how about your? Because you mentioned that it's for your technology similar as the classic cysteine conjugation, but I'm just wondering because you did something different, right? And what's the most challenging step to scale up for your technology?
I think the conjugation is actually a little bit, I wouldn't say difficult to scale up, but that is the relatively more important step for the scaling up because other like purification stuff is very straightforward. Because we are using a metal to block the hinge region, and it's actually a quite intricate balance between like reduction agent and all the materials. So the conjugation part is still the most important part to monitor. But once we reach a certain stage, like gram scale or 10-gram scale, the process is robust and tested. Further scaling up should be fine. And once it reach after the PD stage, the batch-to-batch consistency will be good.
Okay. Okay. Fascinating. Yeah, it's clear that manufacturing actually is kind of martial art as a science. So looking forward, actually the field is moving beyond a single payload ADC to more complex designs. And how can your platform be adapted to create multifunctional conjugate, for example, dual payload ADC? And what are the primary challenges you face in achieving this both chemically and analytically? Dr. Wang, so with the precision like glycan engineering, that seems like a perfect fit. So what are your thoughts?
Yeah, I think I will use the opportunity to also answer your first question, right? So, like for glycatides, glycan side specific antibody drug conjugates, the first generation, we kind of worry about like the larger scale production use complex and glycan. But surprisingly, like Daiichi Sankyo with the license technology for their application, the find out with I think teamed with another Japanese company to do large scale production of the antigraph complex of antiglycans from chicken egg yolks, and then they can modify the glycan and put a drug on it to do the final conjugation, right? So that is the first phase. First generation technology, and the second one is the disaccharide substrate, and Gravity and several other companies like Glycan Link, right? So we've been working on that to scale up. In the laboratory, we can work on multi-gram scale for the production. And for Gravity specifically, we are now working together with another US company to do the scale up for the production of the disaccharide oxazolines. So there are different types of disaccharide oxazolines. Some are much easier to make than others. So we are trying to optimize the production, and hopefully in the near future we'll have the GMP production. And in terms of the multi-specific payloads,
multifunctional conjugate, yeah.
Yeah. So because of this kind of platform technology for conjugation, so you can always have the antibody with the bispecific antibody and do the conjugation, and so that's a kind of flexibility over there for the technology solve. But of course, it will be case by case, right? If you have a specific design there, you may encounter technical issues that have to be optimized, I would say.
Okay. Yeah. Any other speakers want to add more?
Yeah, Cheng. So obviously I talked a little bit about some of the work that we're doing with dual payloads. And I've thought a lot about, what combinations of drugs go together. I think that's a really interesting mental exercise to go through, because you can choose payloads that are complementary in their mechanism of action or synergistic. But then there's, the downside is also toxicity that comes with synergism. Right. So finding the right payloads for a dual payload is challenging, and it's a little bit trial and error at the moment. What I'm really looking forward to is combining cytotoxin with a non-cytotoxic payload, like a molecular glue or an inhibitor of some kind that gives us some benefits without increasing the combined cytotoxicity of that class of drugs.
Yeah, that's pretty good point. Thanks, Marcello.
Sure.
Yinxi, so probably wants to answer some question online.
Sure.
Yeah.
We do receive quite a few questions for our speakers. So I will start with a question for all panelists then. So how amenable are your technologies for multi-payload ADCs? beyond dual payload. It is still early, but there recently was a paper on SPARCS, multi-payload ADCs in bioconjugate chemistry. What analytical challenges do you envision, and how can these challenges be overcome? So it's a question for all panelists.
I think you should start, Cheng.
Yes, Jen, you are a panelist too.
So how amenable are your technology? But I don't have site-specific conjugation technology by myself. So what are the analytical challenges do you envision, and how can this challenging be overcome? Your technology still early. Right. But basically, I think it will take time to really deep dive into all the to really deep dive into because the basically is for the analytical analysis for the ADC, you need to think about using LCMS, you need to think about use heat chromatography, using the reverse phase. And if you have, MMAE, or you have PD, or you have a tubulizing, and then you need to dissect each payload based on their molecular weight. So basically is you have to really try, probably like the into a fab region, or you need to dissect into different pieces. So basically it will be complicated, but it will takes more exercise for different platform to look into. But I probably will let each platforms to expert to answer their technology. Yeah.
Okay, I can go first. So let's talk about something that's reasonable, say 3 payloads. I think the way to do this is to use 3 different chemistries, right? That are that are orthogonal to each other, so that you have control over the sites of conjugation. That gives you some level of control over the numbers and the positions that you're attacking on an antibody, and thus it requires bringing in a technology that is, for example, combining my technology with the AgiCap technology would probably work, because the chemistries could be compatible. So you click on one site, cysteines on the other, and then the non-natural that I use for a third. Otherwise, branched linkers could be used. That could be a really easy way to generate multi-payload ADCs. Not very elegant though, because 3 payloads in one location or a few locations would be disruptive of the structure of the antibody, and then you have other problems.
Yeah, I agree with Manchero's comment. So I think at the end, you will combine maybe 2 different conjugation methods, right? So, but then the question is how far you would like to go, right? If you combine 3 different methods, the production can be problematic, because really a balance thing. And the branched like a kind of linker can be one solution, as Machero mentioned, right? I think that probably the direction to go. Pre-assemble the things ABC on moiety, and then you use the site-specific conjugation method to do it. But the potential downside is you conjugate everything on, in one location. I don't know whether what kind of problem it will cause.
I fully agree with the speakers. I think like even if our WuXiDARx could make dual payload relatively easily, and we could potentially make 3 different payloads all in the hinge region, but that has a big limitation because there are only 8 binding sites, and if you use branch linkers, it will become quite crowded in that region. So for either like dual payload or triple dual payload ADC design, or simply just lower efficacy payload that requires a higher DAR value. I think it's great to combine different conjugation technologies.
Yeah, I also totally agree with other panelists. Yeah, we can provide their ADC to choose the different conjugation site, for example, lysine 248 and 288 for dual payload system. And also we can provide the other type of dual payload ADCAP combined to reduction method, for example, 2 plus 8. And theoretically, we can provide triple payload system to choose lysine 248, 288, and reduction method. But it it would be a problem to the toxic maybe. So it would be balance of the toxicity and the payload number.
Yeah, I think this is a very good chance for all this platform can collaborate, work together to see what's the best, the combination compatibility between each platform.
Yeah.
Because this, cysteine, and also think about if we want to use a two different conjugation method, then if you want to try payload, then you think about you have to use a branch, potentially branched for one of them, and then which side is better or which platform is better for a branched version. So I think there's a lot of more to actually promote more collaboration between each platform here. Yeah.
Thank you, Dr. Lee. Do we have time for one last question? So we have a lot of questions, but I think if we have time, then we could quickly address.
Yeah,
this
we can
last
probably have
question.
quick to answer the question.
Yes.
Yeah.
So this is a question also, I think, to all our panelists. And then I would like to hear the speaker's opinion of the reality of site-specific ADCs potentially being safer or better tolerated than traditional ADCs generated by lysine and cysteine methods. There are some site-specific ADCs that do show better safety profile and or improved efficacy over traditional generated ADCs. But the converse has also been reported. What should one be looking at to ensure site-specific conjugation methods can truly deliver ADCs with higher tolerability? I think let's start with you, Qing.
Oh, sorry, I was looking. Yeah.
Oh, sorry. Yeah, it's the top question.
Oh, okay. Here the top. Oh, this is a tough question. Here, speaker opinion on reality on site-specific conjugation potentially being safer, better tolerated. Yeah, I think basically is the really is asking the therapeutic window, and there and basically there's a lot of complicated factors that contribute to the therapeutic window, like the indication, the target, and also the antibody part. And the amount of the site-specific conjugation play the role that you want to judge based on purely by current clinical data actually is not very fair. And preclinically, we already demonstrate all this evidence that site-specific conjugation that can help to many different perspectives. And to really understand whether how it can be safer or better, we do need to accumulate more and more clinical data to help us understand it. Purely based on one or two report is not fair to answer the question. Yeah.
Yeah, what site-specific conjugation definitely has its own advantage, as you know, right? So the homogeneity, product to manipulate, you have well-defined pharmacokinetic, profile. You can, but it really depends on which side you attach it, right? People have done tremendous studies to compare, which side will be more feasible to attach what payload. So I think like it would be you cannot have a general answer to this question because I think it's still case by case. I think so far, based on most of studies, there are some sites that are better than the other sites. So I think that can be the starting point. And again, like whether the site-specific antibody drug candidate will have a better like a kind of efficacy, this is a totally different question, I would say.
I'll go next. My thoughts on that, first of all, it's a great question because it's a question that we've kind of been asking ourselves for years. Is it worthwhile doing site-specific? And one of the problems in the field is that there are no good comparisons between site-specific and stochastic conjugates. Nothing that doesn't include multiple variables. So it's hard to pinpoint, what limits the therapeutic index, if there's 4 or 5 differences between the stochastic and the site-specific. I think homogeneity, predictability, robustness is really important. And site-specific conjugations give us that. So perhaps we'll be better able to predict how an ADC is going to behave before it gets to the clinic so that we have a better success rate once we are in the clinic. And another thing, what we're seeing with site-specific conjugates, stable conjugates, is that they tend to be more potent and have longer pharmacokinetics. And so when you think about, Raffaele Colombo and Richard Rich published this nice paper looking at toxicities and how ADCs are tolerated based on their payload. Their conclusion was basically that you can only take so much of a given payload, and above that, you see toxicity. If you can make an ADC that has half the DAR and twice the efficacy, then you would expect to see a therapeutic index improvement, right? Because you can dose up and still achieve the same efficacy. And that's something that site-specific conjugates have a promise to achieve. But again, sites are important, linkers are important. There's a lot of variables along the way.
Thank you. Yeah.
Thanks.
Fine.
I'll go next. First, I think potentially, or in the future, maybe in a few years later, I think some types of site-specific conjugation will show clinical benefits. That's my guess, just a personal guess. And I kind of I get the point. Dr. Marcelo mentioned that the review paper showing the highest dose is determined by the payload itself. So the review paper state a fact of the current clinical trial landscape. But personally, I'm not sure if I agree with his conclusion, because in preclinical models, we can make the ADC safer for mice, for monkey. And I understand that different animals are different, but we don't have enough studies of really site-specific conjugations in the clinical to gain the experience and the insight to better design it. But in the future, maybe we will be able to do that.
Yeah, you're absolutely right. And Rafaela's paper didn't cover any site-specific conjugates. So because there are none, right, in the clinic at the moment. So
yeah.
That's a very fair point. The other thing they did not address is increased exposure, right? So they don't account for the amount of exposure of an ADC. So yes, fair point.
Thank you.
Yeah.
Thank you.
Dr. Watanabe?
Yeah. Actually, I presented the comparison of our ADC and stochastic whole ADC. So yeah, I believe the site-specific ADC have the advantage to stochastic ADC in terms of the stability and efficacy and production. But yeah, I think we should look best combination of antibody, linker payload, and so on for each ADC if we consider the ADC development. Yeah.
Thank you. Yeah, thank you again to all our speakers for sharing your expertise, and also to our audience for your active engagement and thoughtful questions. And due to time constraints, we won't be able to address every questions or explore all the details raised today. So, however, we truly appreciate your thoughtful participation and encourage you to continue these valuable discussions beyond this webinar. And thank you. Then that concludes our panel discussion.